Aquaria has been a work in progress for a long time, and what we've been motivated in the team is really this insight that looking at the structure of biological molecules can give, in some cases, enormous insight into the functions that are happening on the molecular level. And it's pretty difficult to understate the importance of the structure of the double helix of DNA. And that was DNA, but we have now many protein structures, each of which have separate functions from the information storage that's in DNA. There's thousands of other molecular functions that are implemented by these molecular machines called proteins that we have. In fact, the number of protein structures that are recorded in the protein data bank has just gone past 100,000 entries, which is really a landmark in human understanding of molecular processes. And this is fantastic. Every one of those structures represents at least 1 man-years of work. But in some sense, the success of structural biology has given us a new problem. When faced with this overwhelming number of structures, it can be difficult for a biologist. And when you go and navigate to one of these structures in the PDB database, which I'm really one of the greatest fans of, but it can be a little bit difficult for a biologist to find the information they need in here, because essentially the protein data bank is designed for and by the structural biology community, which is a small subset of the biologists, those who specialize in structure. And for the typical biologist, when they come to this collection of 100,000 structures and they see these pages that aren't particularly intuitive for them, the initial reaction is to sort of go somewhere else and get back to some other part of the research. And really this idea of trying to make structural biology and the outcome of all that more accessible to the broader biology community is the driving force behind the creation of the Aquaria resource that has just been published in Nature Methods, the current issue. Essentially what we've done is try to reorganize all this information in a way that's more intuitive to a biologist. And we tend to use graphics for that. So I'll just step you through what it looks like before we step into the live demo. So I've just represented here one protein sequence, a fairly typical length, around about 400 amino acids. Can't quite read them all, but they're represented there. And if we consider the relationship now between a sequence and a structure, it's not one-to-one, it's usually one-to-many. There can be many different structures that we can look at when we consider one protein sequence. So this particular protein sequence, one I'm going to use for the first part of the demo, it's p53, a protein very important for all human cancers, or most human cancers. And when we look at the structures that are available for p53, we see that there's a group of structures that match on to the beginning of the sequence. There's a group of nearly 100 structures matching in the middle part of the sequence, which is the region believed to bind to DNA. And then there's other structures at the end of the sequence that are believed to be responsible for the oligomerization of this protein. And so you see structures have been solved in pieces in some cases. In some cases also there are structures available that bring these pieces together in larger aggregates. So there's actually a couple of structures in the data bank that contain 2 of those different domains, 2 of those different structural regions in one structure. So this is sort of how we represent the structures and the relationship to sequence in Aquaria. We use a coloring scheme where the way we represent the sequence and the coloring of that is very similar to the way in which we represent the coloring of the structure. One feature about Aquaria that's I really want to draw your attention to is that every time you look at a structure, by default, we highlight any residue in the structure that's not equal to the sequence that you're interested in. So in this case, we're interested in the p53 human wild-type sequence, and we've gone and looked for structures. And you notice that that last structure that I pulled up there, the one on the bottom, has got a few black marks on it. And if you look at the color legend over to the right, what the black marks tell you is that those are residues that are very different in the structure from in the wild-type sequence. Those are mutations that the crystallographer has actually, in this case, introduced in order to be able to solve that structure. So it's actually, he's got quite a few black marks, so it's quite different from the wild-type sequence. For p53 currently, it's around about 120 structures are available that you can look at. Every one of them generally will tell you some particular story about the function of p53. So you don't necessarily need to look at them all, but the idea of Aquaria is to create an overview where you can see graphically where all those structures fit onto your sequence and to make each of them accessible. So let's switch to the demo side of things. So to find a protein in Aquaria is quite easy. You just type in the name of your protein. In this case, I've already loaded it to p53 to type in that name in the field here. So we tried for Google-like simplicity with just one field, but biology, of course, is complex, and so there's one additional complexity we have to manage, which is that there's p53 available for different species, so you also need to specify a species. We default to human, but you can change that by typing in your favorite species there. So having loaded the structure, loaded the sequence that we're interested in, Aquaria then displays some basic information about the sequence. And probably the first most useful thing about this view is this visualization down the bottom that is essentially a concise visual summary of everything that's currently known about the structure of p53. It's really all organized here on one page, and you can just come back, bookmark this page, and come back there and check and see what's going on. So you see that there is no single structure that spans the entire sequence. Right? That's just not known currently. We don't have a structure for that. And indeed, there's no structure matching certain parts of the sequence, like here, there's a complete region that's missing structurally, and also here there's a region that's missing structurally. And notice that this structure that I pointed out before, this group of structures that spans two of these domains, it seems to be the best match. And so this is the thing about Aquaria. We, by default, try to show you the structure that we believe is the best match. And we use a very simple algorithm for that. We choose the structure that has the largest number of identical residues when aligned onto the full-length sequence that you're interested in. Okay? And so that's why, by default, we choose this structure, because it matches two of the domains. So it would seem to be the best structure to look at initially. And so the structure is shown up here. Notice that there's another molecule going on in the background, because when you look at a structure, sometimes there can be multiple proteins or even DNA, other macromolecules in the system. Aquaria is designed to sort of show you the overview, but to focus on one protein at a time, the one that's most related to the protein that you're interested in. You can easily move to another one just by clicking on it. Clicking on it will make that the center of rotation. And will change the view. If that was a different protein, it would actually change all the information and tell you information about that protein. In this case, this is actually a dimer of p53, so they're both the same protein. So that's our way of dealing with the cases where you have a very large number of molecules. You focus on one at a time and provide an easy way of navigating around those large structures. So let's look in detail at this. As I mentioned, there's this color scheme that shows you every residue that's different in that structure from the wild-type p53 sequence. And we see quite a lot of them. In fact, there's about 19 amino acid substitutions that have occurred, that crystallographers have introduced in order to be able to solve the structure. You can find out more by reading the abstract about that particular PDB structure. Notice also that I mentioned there's this gap from around about residue 300 to 320, around about 20 amino acids, that there's actually no structure in that region. And this structure spans that region, but there's a gap. So what the crystallographers have done is they've produced a construct that fuses together two domains, skipping out 20 residues that are normally there in the wild-type sequence. So this is quite different from normal p53. And for all those reasons, we might decide that this may not be the best one to look at, even though it has two domains and spans a relatively large part of the sequence. We see there's a lot of substitutions have been made, and perhaps we should look at some of these others. So all the structures are ranked in order of their sequence identity to the wild-type sequence. And we see there's a group of structures up here in the DNA binding domain that have 100% identity. There's 86 of them, and I can click on that number, and what this will do is open up a tree view, and that shows us, it's a way of taking those 86 structures. We know they all match in that region of the sequence, but 86 is still a lot of things to deal with, so we've broken them down, and you can see that of the 86, there's around 45 that consist of a single molecule of p53 just by itself, no other macromolecules in the system. And then there's some that bind to a range of different macromolecules, including, for example, DNA. We've got here, for example, DNA dimer binding to a p53 tetramer. That's actually believed to be one of the most physiologically important states of p53, so we'll have a look at that, but just to show you how to scroll up and down the list, essentially we've summarized all the information about those 86 into a fairly concise view. Let's look at that structure of the p53 tetramer bound to DNA dimer. There's 3 different PDB files there. I can just mouse over them to find out more information about them, so I'll just choose one for now. So what this is now going to do is load that structure into view, and as we saw before, there's now more molecules in the system. If I want to sort of zoom back and look at the whole thing, There's a couple of ways to do it, but the simplest way is perhaps just to double-click on the background. That kind of zooms to everything. And now you see the attachment of the 4 copies of p53 and the DNA dimer there. And again, just click on one part of p53, and that'll focus on that particular monomer and enable you to view details about it. So this is quite nice. We've organized the information in a useful way, given that useful summary of every structure that's related to p53 and a pretty nice way of navigating through it. But there's more, and that's available in what we call the Features tab. So what we've done here is bring in features from a number of different resources. These are features of the p53 sequence, not of the structure. And there's quite a number of features here, actually over 1,000 in this case. And they tell us a range of things. So for example, this feature set tells us different regions of the sequence that are known to interact with other molecules. And what I can do is just click on a feature lane, and it'll take that lane and use it as a coloring scheme for the molecule. So I can see which parts of p53 bind to those different proteins. For p53, probably one of the more useful feature sets is down here. These are the the genetic variations of p53. Essentially, each of these mutations is believed to be responsible for a cancer. So they've been all derived from tumor tissues. And the way we've ranked them actually shows you the residues that have the most number of distinct mutations in p53. And we can click on that feature lane, map these most sensitive residues onto p53, and then shows us where those most sensitive residues occur. And by looking at the organization and the spatial clustering of groups of mutations, we can sometimes learn new things about the functionality of this molecule p53. So this was a case in p53 where we every structure we've looked at up until now has been determined using the wild-type p53 sequence. But many of the proteins you're interested in probably don't have a structure that's available for them, but they do have a structure available for something similar. And let's see how Aquaria handles cases like that. So I'm going to choose a protein here that's been worked at, actually was discovered by a former member of the garden called Group 4. The Group 4 is a transporter protein that lives in our fat cells and is responsible for the uptake of glucose. So what we see now, first thing you notice in the view is that there's a lot of residues that are dark. And that's because the structure we're looking at now is actually not the structure of group 4, the sequence you're really interested in. You can always tell in Aquaria what you're looking at. It's in the very top bar. It tells us what's going on. We're looking at the structure of group, we're looking at the group 4 sequence aligned onto, in fact, this is the group 1 structure, which has 68% sequence identity. Okay, so this is the closest matching structure for group 4. It's a distinct protein called group 1, and that's 68% identical in the sequence, which is pretty good. It's quite close. And all of the residues that are different are, as I said, annotated using that darkened coloring, so we're very clear about where the differences lie. Here, the features can provide us with a real sense of orientation. So there's a feature set down here, for example, that I can turn on, and it'll annotate the parts of that protein that are occurring in the cytoplasm versus those that occur extracellularly. So this is a small example of how turning on these feature sets can give you an insight into function. And more insight can be gained by looking at all of the structures. This is really one of the advantages of Aquaria, is for every one of the sequences we've determined not just the best match, but all available matches in the PDB. And in this case, for the group 4 sequence, we have around about 70 matches. Some of them have a very low sequence identity. You can see it goes down to very low values, but it can indeed still be worth looking at those lower matches. We've spent a lot of time in optimizing the algorithm, and we're fairly confident about it. Our next speaker, Andrea, will be talking about the methodology behind the matching that we've done. But essentially, by looking at different structures, you're able to see information about the different functional states of the protein, or of proteins similar to the one you're interested in. And in this case, the structure that we're looking at is a molecule that allows essentially sugar to diffuse in from the top to the bottom. And if you look through those set of structures, you see some that are in what we call the outward open state, where the sugar from outside binds. And there are other structures that are very clearly in what's called the inside open state, where the sugar then is released inside the cell. So again, by looking at all of the structures that are available, similar to your sequence, you can learn about the molecular mechanisms of a given protein. So now we're going to try to bring in Andrea. Andrea, if you could try to connect, and we'll try to call you in. So Andrea's my... Hi, Andrea. You're there.
Hey, I'm here.
Good.
I'll just start screen sharing and my presentation in just a moment.
Good. So Andrea has developed the database underlying Aquaria. This is actually
can you see my presentation?
Two master's theses in the way of doing this. And so she's going to present how the database was derived and what some things we can do with it. Okay, Andrea, you ready?
Yes, I'm fine. Can you see my slides?
Yes, we can.
Okay, cool. Okay, so hi everyone. I can't see you now anymore because my screen doesn't show you. I'm going to talk a bit about the database behind Aquaria, which we call PSSH2, and explain about how did we derive it and why it's called like that. And in the end, I'm also going to talk a bit about mapping external features onto Aquaria, which I think is one of the really cool things about Aquaria too. Okay, so as Sean mentioned already, it's not always easy to find a structure. If you consider that what I'm showing here is kind of old sequences that would exist in Swiss prots, only about 4% have any structural information at all directly made for that sequence. However, since 1991, there was the idea that by homology you could extend the amount of structural information because you can infer structure based on the known structures. And in 1991, the Sander group published a paper about the HSSP database that belonged to the overall family of DSSP, FSSP, and for every PDB structure, it listed all the sequences where you could infer the overall fold based on the homology. The HSSP database was based on the program MaxHom, and that was, to my knowledge, one of the first profile-based alignment tools, and it was pretty good in the alignment quality. At that time, there were about 20,000 sequences in HSSP, and they covered about 34% of SwissProt. So when we decided to build the predecessor to Aquaria, we decided that probably the HSSP database is a good place to start with. However, in HSSP, everything was organized by PDB structure, and now we were interested in listing all matching structures for a sequence. Therefore, we had to turn the organization around, and therefore we also turned the name around. So we now call our database PSSH. And from 2003 on, we maintained that database. When we published it in 2003, we had about 400,000 sequences in there. That was about 48% of UniProt, and it covered 25,000 PDB chains. However, the databases, sequence and structure databases continue to grow, and in 2010 we had more than 500,000 sequences in SwissProt alone. So SwissProt is the curated part of UniProt, or well-curated part of UniProt, and we had more than 60,000 PDB chains. And this more or less reached the limits of the whole MaxHom and HSSP method, and we needed to look for a faster method. And we decided to call that, therefore, then upgrade our new database name to PSSH2. So what did we look for? Well, let's go back. Basically, we wanted a profile-based alignment again, and therefore there were two options. Basically, we could use SciBlast, There was a new method called HHBlitz, which is a sped up version of the HHSearch program that existed before. The idea behind HHBlitz is that you do a HMM against HMM search. So in SciClass, the principle is that you build up a profile for the sequence you're searching with, whereas in HHBlitz, you have profiles on both sides. You have profiles in the database you search against, and you have profiles for the sequence you search with. So it can become even more powerful. And that it can become so powerful was shown in the CAST competition for homology modeling. In 2010, the two predictors, HHPRED-A and HHPRED-B, which are both more or less based on HHBlitz, came out first. In 2012, they did not come out first anymore. However, if you look at the results closely, you see that the methods that come out better either use HHBlitz in some way, or they're at least 370 times lower. So this showed that HHBlitz is a pretty powerful method. So we were confident that this would work, but we didn't just want to trust that, but also validated the method. And that was part of the master and bachelor thesis that Sean mentioned. What we did is to use a database called COPS, which is based on the structure alignment tool TopMatch. And in there, you can find all the PDB structures that have at least 30% structural overlap with any query structure. They form groups. It's a bit like SCOP and CAFT databases, but the difference is that this is based only on structure alignment. No sequence alignment at all goes into that database. That's why we decided to validate against that, because we just wanted to look for really structural similarity. Okay, there are true positives and false positive sets were defined by true positives had to have 30% structural overlap, and everything that didn't have that was considered a false positive. Then we compared what is the true positive rate and the false positive rate if we use HHBlitz or SciClass to search, to take one structure and search it against all the PDB structures. And as you can see, in the region where you have a very low false positive rate, these are the high confidence alignments. The SciClass and the HHBlitz curve is pretty much similar. However, if you go to a bit lower, a bit higher false positive rate, then you get more true positives at any false positive rate for HHBlitz. So HHBlitz can give you more recall. And in order to tune our method and decide at what level do we really show structures, we also looked at what e-value do we receive, what recall and what precision. And we decided to cut at an e-value of 10 to the minus 10, because there you already have a pretty high precision of about 70%. And you have a really high recall of over 80%. Okay, so this is H2 database built. What we do is that we take the UniProt database from the HHBlitz suite. We get the database of profiles called UniProt 20. Then we take all SwissProt sequences and scan them against the UniProt 20 database, which gives us a HMM for every single SwissProt entry. Also, we take every chain from the PDB and scan it against UniProt 20, which gives us a database of HMMs for every PDB chain, and we call this PDB full. In the HH split suite, you get a redundancy reduced PDB database, but we really want to include every PDB chain. Therefore, we built our own PDB full. Then we can take the switchboard HMMs and scan them against the PDB full and get our PSSH database. And with that, we can get from 4% sequences that have some easily accessible structural information to 87% of switchboard sequences that have structural information. That does not mean that the whole sequence would be covered, but that for 87% of the sequences you can at least find some structural information. At least part of the sequence will be covered. For any single residue, you have a chance that of about 50% that you have structural information. And at the time we published Aquaria, there were 46 million sequence to structure alignments in the database, and 85 structures per protein. Okay, to the other thing I mentioned, mapping protein features. I come from a lab of Burkhard Rost, where we're interested in predicting loads of protein features from the sequence alone. And one of the things we predict are the effects of point mutations. And what we often do is to look at every possible point mutation. It's like an alanine scan, but we don't just scan against alanine, but every possible residue. And we see whether whether this mutation could be neutral or detrimental. What you see colored in green would be neutral mutations, and red would be some that would change the function. Now, an overview like the one you can see in the slide here is not extremely informative per se, or we could enhance that. And the cool thing about Aquaria is that if you modify the URL you see on top of the browser a little, you can give it another parameter called features, and then you can give it a URL where it can load the features from. It's all described in the feature API. And what this results in, that in the features tab in Aquaria, you see another lane which gives you the annotation you have loaded. And then you can map that onto the structure, and suddenly these effects of point mutations start to make a lot more sense, and you can ask more questions. So the sense it makes in this example, I'm showing, by the way, GLUT4 again, which Sean just mentioned. What you see here is that the mutations predicted to be neutral overall are mainly on the rim of the protein, so outside, whereas the mutations predicted to be to change function are for a large part pretty close to the binding site, or in general inside the protein. So this whole pattern starts to make sense if you look at it in 3D. Okay, finally, a word about the amount of data we're dealing with here. I mentioned it briefly just in the before mentioning the features. Currently, in January 2015, there are about 460,000 non-redundant switchboard sequences. There are about 72,000 non-redundant PDB chains. Non-redundant means that I count things as redundant if all the residues are the same. And this results in 48 million alignments. So you have more than 100 alignments per protein now. And this is more than 11 gigabytes of alignment data. And of course, we are grateful that we got help for hosting this, and I think Sean is now going to take over and hand over to our next speaker.
Okay, thank you very much, Andrea. That was in case the demo didn't work. So that's the feature API, and this is really important for institutes like Garvan. This enables us to go beyond the limitations of what's in the sort of standard Aquaria, and for Garvan, for example, to take information about genetic variations amongst a cohort or a group of patients, define those into a JSON file, which is a very standard format in bioinformatics, host them somewhere, and then just map them onto any structure. So it's a really powerful and useful feature of Aquaria that's quite new, and play with it and get back in contact with us, because it's still got perhaps a few bugs, but we'd like to iron them out. So it's Andrea sort of outlined the sort of the problem that we have. If we want to deliver Aquaria to you, we've got 46 million structural models, many gigabytes of data. And how do we make this accessible to people? So obviously you see we've lavished a lot of attention on the design of the user interface to make it all easy to access. But then when we came to the hosting issue, we had a quandary. We could host it here in Australia at the institutes we work at, but most of the people who are going to use it are going to be international scientists, I guess primarily US and Europe and so forth. And we really wanted something that had high quality of uptime and so forth, a server we could just rely on just sort of being there. And that's when we approached Amazon about this. And in fact, the main Aquaria server that you use, Aquaria AWS, is hosted in North Carolina currently. And now I'd like to invite on the stage Neil Jackson from Amazon to say a few words about the hosting of Aquaria.
Thank you very much, Sean. I won't keep you very long, everyone, but to Dr. Sean O'Donoghue, the team at Garvan, Andrea Schafferhans, I'm hoping I got your name right, Andrea, those present here today, and also everyone listening in through Google Hangout. Wow, I mean, I'm blown away by this sort of work, so I think we're in awe, and we're honoured to be involved in supporting the Aquaria project, and we're delighted that today marks the announcement of its broad availability as a web-based hosted service by AWS. We'd like to acknowledge the leadership shown by Dr. O'Donoghue and his team, and hope that this is a resounding success to all life scientists around the world. So through our scientific computing team at AWS, aptly named Psycho, led by Jamie Kinney in the US, and also by Brendan Bouffler here in Australia. If you haven't met Brendan, he's sitting in the front row. We'd like to acknowledge the work, and Amazon Web Services supports numerous groundbreaking science projects such as Aquaria, and makes available large data sets such as the 1000 Genome Project to help scientists and researchers collaborate across the globe. We really hope that Aquaria is a roaring success, and we look forward to a continued relationship with Dr. O'Donoghue and the Garvan Institute for many years to come. So I'd like to thank you for the opportunity to be involved in this piece of work, and we hope to be talking again in the future with Amazon and the team. Thank you very much.
So I'd like to extend my thanks to Amazon, who have provided hosting of this service with a grant. So not only the service, but actually the underlying database that Andrea mentioned, PSH2. You can access and use this data not only through the web portal, but if you're a bioinformatician, you can access that data programmatically. This is all described on the Aquaria help. So finally, our last speaker is Chris Hemang, who's an animator who's been using Aquaria over the last 6 to 8 months, producing a new animation that's actually going to be featured tomorrow in Canberra at an event hosted with Australia's chief scientist. And he's going to show you a sneak preview of this animation that no one else has seen. And he's going to explain how he's used Aquaria to piece together the puzzle that he's been working on, which is a protein that's key involved in the Alzheimer's disease. And now I have to switch over to your presentation. Here. And then just go.
Okay. Thank you, Sean, and thank you for inviting me here to speak to you today. So I'm only going to talk to you briefly, and from a slightly different point of view. I'm an animator, as Sean has said, so I make videos for the general public. This is a video that's going to be released tomorrow, as Sean said. It's actually going to be called Alzheimer's Enigma, and it features some complex biology about protein dysfunction that occurs during Alzheimer's disease. But I do just want to highlight that the purpose of my use of Aquaria is for animation and for public outreach, so it's not necessarily for scientific inquiry. So just to give you a quick recap of what Alzheimer's disease is, basically as the disease progresses, the brain tissue shrinks and dies over the course of decades, usually, and it's most common in senior 65 age and up people. And we see that the cerebral cortex has extreme atrophy, and also in hippocampus regions of the brain. One of the underlying features of the disease seems to be this misprocessing of proteins. So this is a representation of the cell membrane on the neurons of cells, and this is a protein called APP, highlighted in red here. This protein is recycled like many different proteins in the body, but it has two different pathways of recycling. One of them, called non-amyloidogenic here, is basically seems to be if it's broken down with one set of enzymes, we don't get these plaques that occur in the disease. But if it's broken down in the amyloidogenic pathway, what happens is we get an accumulation of this small protein fragment, this one here called AB, or amyloid beta. So if this set of enzymes on the right here is used, it can lead to accumulations of the protein in cells, and that seems to be associated with the disease. So this is what just a diagram of there's different ways that these accumulations can occur through oligomers, through fibrils, and this formation of these large plaques, which are the size of cells. So there's been some attempts by scientists to model the structure of this protein because it seems to be related to the disease, and it seems to be quite critical. But it's actually been difficult because they haven't been able to resolve the structure of the entire protein. Similarly to what Sean was mentioning, this is often a problem with proteins, especially transmembrane proteins like APP. That reside inside the membrane of the cells. But there have been attempts, and there have been certain sections that have been resolved. So what I found in doing the animation, I wanted to communicate some of the biology about how this protein is processed and how it might relate to the disease. But to do that, we needed to construct a model of the overall structure of this protein so that we could represent it, and that's where Aquaria came in. Yeah, so I'll switch over now to... Oops. So what I actually did was, in order to create a reasonable model of what this APP protein looks like, I assembled all the structures we could find from Aquaria. So as you can see here, like the other proteins Sean was showing, we don't have a completely resolved structure of the entire protein sequence. We have fragmentary information. And actually, as you can see, this piece here has 150. There's a large number of structures throughout the different regions, but perhaps because it's a transmembrane protein, it's been difficult to get an entire structure. Using Aquaria, though, we were able to piece together a number of different fragments in a reasonable fashion for the purpose of the animation to construct a model. Also, using the features view, you can identify the region that is transmembrane. So we can see that only a small portion of the membrane of the protein actually sits inside the membrane. The vast majority sits outside of the cells. This is one of the central structures from within. This is the largest structural element that we have access to. And we have access to a number of structures within the transmembrane region as well. I'll show you what the resultant video looks like. So the animation starts by showing you the neurons of the cells, and then we zoom in to the molecular surface of the cells, and this is the structure that we generated for APP. So this is the portion that sits outside the cell. In the animation, we actually cut through the membrane here so that you can see. So we wanted to expose what the entire structure looks like. So this is things getting in the way. This is the transmembrane region there, and this is the, as you can see, the large portion of the protein that resides outside the cell. And there's a small anchoring component within the cell as well. So unfortunately, you don't get to see the whole animation today, but that's a little sneak preview there. But the video will be available tomorrow, so on YouTube. bizbit.org/plus is the place to go. Yeah. Where's my... area. So yeah, that's basically it. I just want to say there's a large number of people behind creating these animations. Thanks especially to Sean Donahue and Kate Patterson, who put in the grant application to get funding, and Inspiring Australian and CSIRO, who funded and supported this project. And so the video will be called Alzheimer's Enigma. Go to visbia.org/plus, and you'll be able to see it as of tomorrow. Thank you.
So I hope that gave some people imagination and ideas about how one can use Aquaria. And really what he showed was how he pieced together the whole story of the breakdown of the different parts of the protein by looking back and forward at the structure view and then at the features available for the sequence. So in the remaining moments, I just wanted to cover some, I'll get to some practical points, but first to show something a little bit on the fun side. So this is actually a controller we built nearly a year ago now. So if you are an owner of a thing called a Leap Motion, which costs about $100, you can plug it into a USB port, and this will actually now work with the Aquaria website. So you just go to Instruction Aquaria, and if you don't want to have to learn the mouse controls to rotating and translating, you can actually just sort of virtually hold the molecule, pretend the molecule is your hand, and rotate it around. And fun to play with. But a few practical notes. So the main version of Aquaria is actually a Java applet, and currently it runs best in Firefox if you're on Windows, and either Firefox or Safari if you're on a Mac, and of course Firefox if you're running on Linux. If you do run on Chrome, I'll just show you what happens. First of all, you get a message. See the message at the top that says you're going to have limited functionality. Because what happens in the case of Chrome, we do actually have a viewer, but it's not the Java applet. Instead, it's essentially a preview of a resource that we're developing, which will be a JavaScript-based version of the whole thing. Just reload this. And in the future, we are going to move towards JavaScript for the molecular viewer part, but currently this view is a little bit preliminary. It loads actually a lot faster than the Java applet, but it's got a lot more limited functionality. And let me just also encourage you to click on the help button. So we have not only spent a lot of work to make Aquaria generally useful, but also to make the help useful. So if the help button's up there, and if you click on it, you get this sort of overlay telling you what the different parts of the screen do. And in particular, we've got instructions for getting it installed. The first barrier you'll have is usually installing the app that you need the latest Java. And if you run into problems, check out the help pages there on your environment-specific help page. There's a quick reference card that actually gives you a one PDF, one page, or one double-sided page summary of all of the commands in Aquaria in the 3D viewer control. You don't need to learn them all, but they're all there for easy access if you want to look them up. And then the API. So for the bioinformaticians in the room, this is probably the most interesting thing. You can do lots of things with Aquaria. You can pull data from Aquaria in different ways. And as Andrea demonstrated, you can actually add your own data to Aquaria in various ways. You can even directly access the MySQL table, holding PSH2 directly from Amazon. So do use the help. So if you've got any questions, in a couple of weeks we're going to be hosting a Reddit Ask Me Anything. So we'll have the precise details on the same page that you're looking at, the aquaria.ws/launch page. We'll put everything up there. So really, just want to encourage you to try the service out. I mean, just type in the name of your favorite protein. It's fairly responsive. We've really lavished a lot of attention, as mentioned, on making the whole thing easy to use. I'd encourage you to use it and start exploring this interesting world of protein structures that are out there. There's some bizarre and wonderful structures out there, and you can learn a lot about the molecular functions that are happening. So with that, I'd like to close the event, and thank you very much for your attention.